Hello, welcome to the lecture, and I'm Thomas Ming-Shui Chang. I've been working in artificial cell for more than 60 years. It's a pleasure to briefly summarize this area for you. First, let's ask the question: is it possible to prepare artificial cell consisting of monothin membrane with inside separated from outside, but also outside cannot enter? Small molecule, however, can equilibrate very quickly to be acted on by inside and come out. The first simple one prepared consists of hemoglobin inside artificial cell. It binds oxygen and releases oxygen in a way very similar to red blood cell. Now, after medical school, I went back to work full-time and become more ambitious and asked the question: red blood cell consists of membrane enclosing hemoglobin and lots of enzymes. Is it possible to prepare the same artificial cell except changing the membrane to an artificial membrane? These are the artificial red blood cells that are exactly the same in content as red blood cell, except the membrane is now nylon and cross-linked membrane protein. The hemoglobin and red blood cell enzyme is retained inside, separated from outside, but substrate can go in, acted on, and then come out. These are very thin membranes, so when you break it, it becomes shown here as ultra-thin membrane here. It's very thin and very permeable, so if you put it in hypertonic solution, it shrinks as water comes out, and if you put it back in hypertonic solution, it retains its shape as water re-enters. You can change the membrane, making it with lipid complex polyhemoglobin. You can also put in passive transport mechanism. You can also put in active transport mechanism. So therefore the answer is yes, we can prepare a complete artificial red blood cell. But can we use artificial to innovate, to translate? So let's ask the question: can we vary the content, size, and configuration? The beginning attempt at innovation is done whereby artificial cell of nanothin membrane can be entrapped with cells, hemoglobin, enzyme, magnetic material, adsorbent, and other things. It's separated from outside, molecule can go in, acted upon inside, and then come out. Now, going further now, at present, you can in fact put in all sorts of material, artificial, biological, or active material, as you can see. So there's unlimited content. And you can also vary the configuration from micron dimension under different names. People like to give different names when they extend it for different uses. You can make it macro when you put in cells, and adsorbent. You can make nano dimension. People call it nanoparticle, nanocapsule, liposome, and others. You can make it also in the form of nano biotechnological soluble complex, for example. In my 1972 monograph on artificial cell, I emphasized that artificial cell is just an idea. Much physical configuration are possible. Indeed, in the last 10 years, there has been extensive increase in configuration as extension, improvement of the idea are being made. within a few more years, it's expected that much more configuration would be possible as the area is extended and improved. Thus, indeed, content, cell size, configuration can be varied quite extensively. But the question is, what can you do with all these? This is an example of the possible uses. I'll let you look at it. Microdialyzer, drug delivery, blood substitute, biotherapeutic, vaccine, cancer therapy, immunotherapy, enzyme gene therapy, cell therapy, biotechnology, nanotechnology, regenerative medicine, agriculture industry, nanocomputer, nanorobotic, nanosensors. So let's take some of these as example to discuss. This is the micro or nano dialyzer bioreactor based on nanotechnology and nanomedicine. Let's look at an artificial kidney. Complex, useful machine, large, expensive. Let's ask the question, 10 cc of artificial cell with 10 nanometer thin membrane, how does it compare to a whole artificial kidney as far as mass transfer is concerned? Let's look at mass transfer. Artificial kidney is 1, and the artificial cell of different diameter is shown here, and the mass transfer is shown here. When it's about 100 micron diameter, the transport rate is 100 times. What it means is that things can go through the artificial cell very quickly. However, with 10 cc, there's very limited amount of diffusion can take place. So as a result, we can put in different things to make these into microdialyzer / bioreactor. Therefore, things going in would not be equilibrated because they could be removed or acted on by the content. Let's look at adsorbent. Adsorbent, you can put a membrane around it, 10 nanometer thick. This way, permeant toxic substance can go in, particulate doesn't come out, cell cannot be removed. But so let's look at this in 70 gram artificial cell with adsorbent, and each of these is shown here. This column is schematic with millions of artificial cells. A patient, if you put in blood, the blood would go through, cleanse, and return to the patient. Let's look at how it works. The artificial cell compared to artificial kidney, 70 grams. This is a patient admitted with 3 times the level of the sleeping pill, comatose, in shock, the heart stopped a number of times. When you put artificial kidney, it doesn't remove the drug, so they asked me to go and use my artificial hemoperfusion. When I put it on the patient recover blood pressure and reflex and fully recover. When we analyze the drug level, it shows that it comes down very quickly. There was also an infant that accidentally given overdose of theophylline for asthma, and the level was very high and it could cause irreversible brain damage. As a result, they asked me to put this on the patient. The level came down very quickly and the patient recovered. I have done about 11 of these and it all worked well and FDA approved it. We carry out clinical trial on chronic renal failure patient lasting up to 8 months, and this approach was approved by FDA for device. This is a 2017 book showing that it's now being used around the world for treating poisoning, endotoxin removal, kidney liver failure, immunological diseases, removal of unwanted cells, and more recently, Jeffron has used it to treat COVID-19 cytokine storm, resulting in many people recovering. The first one company started up in Montreal in 1980, 2010, Jeffron in China is producing different types of hemoperfusion devices and is now the third largest Chinese medical. Let's go to the next topic, blood substitute based on nanotechnology and nanobiotherapeutic. This is a rather complicated subject. I will just go briefly. As we mentioned before, we can make artificial red blood cell that's exactly the same content of red blood cell except with artificial membrane. We can make it down to 1 micron. Red blood cell is 7 micron, but even then it's still too big to circulate for enough length of time. This led to the start of nanobiotechnology to make this very small. There are different basic methods of preparing artificial cell. One of this is by imvemtion method to form artificial cell with a membrane. Another approach is nanobiotechnology based on the cross-linking of hemoglobin into polyhemoglobin. Another way is by conjugation, whereby hemoglobin is attached to polymer in the form of polyamine or dextran, or most importantly is polyethylene glycol, PEG. This is done as follows. Hemoglobin and enzymes in free solution are example cross-linked into polyhemoglobin, soluble polyhemoglobin, and the cross-linking agent could be diacid or glutaraldehyde. Glutaraldehyde is the one that's most commonly used. There was no initial interest in these because people perhaps it's a bit too advanced, too early. So it's until 1989 when HIV contaminated blood come. There's nothing to help the patient, and many patients got infected with HIV from donor blood. At that time only that people around the world, as shown here, actively start to investigate different types polyhemoglobin, conjugate hemoglobin, crossing textrameric hemoglobin, also recombinant and bioengineered hemoglobin, and also encapsulated hemoglobin. Let's take one example that's most extensively developed. This is polyhemoglobin crossing with glutaraldehyde. BioPure used mobile hemoglobin to cross-link with glutaraldehyde, resulting in glutamate 250. They did clinical trial in patients on orthopedic surgery, and it was approved in 2002 in South Africa and Russia to avoid the use of HIV contaminated donor blood. So, original aim reached to avoid use of HIV donor blood. What about its use in other conditions? Arterial obstruction, for instance, heart attack, stroke, can quite often be caused by obstruction due to atherosclerotic plaque. In this situation, red blood cell particle cannot go through, result in ischemia. Can we use hemoglobin, polyhemoglobin solution? Because it's a solution, so it should be able to pass through. Yes, it does pass through, and it does supply oxygen. The only trouble is that it causes oxygen then radical release and ischemia reperfusion injury, resulting in polyhemoglobin superoxide dismutase and catalase. And we look at the effect on brain edema in a case of hemorrhagic shock and ischemic cerebral ischemia. The results shown here: control, polyhemoglobin, polymer with the enzyme, showing that it doesn't cause edema. However, you have to cross-link the enzyme. If you don't cross-link it, the enzyme will remove very quickly. Because of this basic research, a lot of other people start to look at different ways to remove oxygen radical. Another one is in severe blood loss, in emergency, accident, natural or man-made disaster. For instance, donor blood in emergency. It has blood group. And because of the blood group, you have to cross-match it before you can give it to the patient. For instance, you have to wait for the ambulance, bring it to the hospital to cross-match before you can give it to the patient, resulting in much delay. Now, polyhemoglobin has no blood group, so you can give it right away in the ambulance without delay of going back to the hospital. Can you do this? Well, polyhemoglobin polyhem from Northfield has been clinically tried in 714 patients. What they do is they give this right in the ambulance, polyhem, with control group giving saline. What they found is that when they reach the hospital, no need for blood transfusion. In the case of polyhem, in saline control, they need blood half an hour, quickly. However, there is 3% non-fatal cardiac side effect compared to 0.6% in the control group. However, looking at risk-benefit, if there is no blood available, you only have saline, then lots of patients will have died. But and then you can give polyhemoglobin, saving lots of life. But if you have blood available, you may want to solve the problem of cardiac side effect for more widespread use. Trosted et al. Found that myocardial ischemia is related to the increase in tissue oxygen, tissue PCO2 related to myocardial ischemia. This being the case, we look into a way to solve the problem. For instance, red blood cell contain hemoglobin and enzyme. One of these is carbonate anhydrase that is responsible for removing CO2. So what we did was to cross-link this together into a nanotherapeutic red blood cell containing hemoglobin cross-linked to 3 enzymes. What is good is that you can increase the enzyme to 6 times that of red blood cell. You can also therefore increase the function up to 6 times. This shows the ability to increase the enzyme concentration up to 6 times that of red blood cell for all 3 enzymes. So let's look at what happened in animal study. This is animal study with rats removed 2/3 of total blood volume and in shock for 90 minutes. We follow the tissue PCO2 and it keeps increasing with time. We then infuse different type of solution. As you can see here, the one with the polyhemoglobin and the 3 enzyme is better than blood, is even better than polyhemoglobin. So since increase in PCO2 is related to increase in heart ischemia, since this is much more efficient in removing PCO2, so let's see what happens. If you look at the recovery of the heart from hemorrhagic shock and ischemia, let's look at using the recovery of ST elevation of ECG. Control saline, no recovery. Polyhemoglobin, 60% recovery. Blood, 40% recovery. This new nanobiotherapeutic recover to 30% of normal. Thus, recovery better than parahemoglobin, even better than blood. If you look at histology of the heart, the blood causes some damage, polyhemoglobin causes more damage, and the new nanobiotherapeutic has no injury signal. We then look into the long-term safety and immunological effect. You're injecting every 4 weeks for 5%, and then 5th week exchange 30%. Look at blood pressure, look at biochemistry, look at antibody, different type of thing. In particular, we also look at anaphylactic shock, measuring both the histamine and tryptase, showing that there's no anaphylactic changes. As a result, the details has been published by Gu and Chang in 2018, and you can refer to it. There's long-term safety, no immunological problem. The result is rather complicated, but it's shown here. You'll refer to the publication. In summary, there's no significant difference from control in body weight, biochemistry, blood pressure, in antibody immunology, and in the no anaphylactic reaction using tryptase analysis. As a result, the long-term safety is demonstrated and there's no immunological problem. The temperature stability is also important at the fridge, 4 degrees, and room temperature. Donor blood, 42 days, the fridge, 1 day, room temperature. Polyhemoglobin, SOD with the enzyme, the 3 enzyme in solution, 320 days in fridge, 51 days in room temperature. But if you lyophilize it, you have more than 320 days, and you can be 220 days in room temperature. You can easily redissolve it before use. Heat sterilization, 70 degrees for 2 hours. The liquid form cannot stand it, but the lyophilized form can withstand it. Cost of the enzyme is also important. For 500 cc of the nanobiotherapeutic, it costs impossibly $250,000 using commercial pure enzyme. But if you use our enzyme extraction method to extract the enzyme from red blood cell, it only costs a fraction of this. So we have the soluble nanohemoglobin with functional membrane. You can also prepare particularly nano-red blood cell with non-functional membrane, PEG, PLL, or PEG lipid membrane. For more detail, you are referred to the book on nanobiotherapeutic-based blood substitute. Let's look at enzyme gene therapy. Enzyme inside artificial cell doesn't come out, is excluded from immunological action outside. This solute substrate can go in, be converted to product, and come out. First report, use it for urea removal, use it in congenital hereditary diseases, use for cancer, use for bilirubin removal, and use for multi-enzyme with cofactor recycling. Another one is to inject artificial cell containing enzyme for hereditary enzyme defect. For instance, for academic mice, the elevated level is decreased by injection of artificial cell. In this case, hereditary enzyme defect require long-term treatment for the remainder of the life, so repeated injection of artificial cell will not be practicable, but this could be solved by solution is to use oral injection. For instance, after oral injection, it moves down intestinal tract and is then excreted after it completed its function. Oral administration. For instance, in Leishman disease, it's an enzyme defect that's very rare, and the Montreal Children's Hospital contact me if there's something we can do. It's the trouble with hyperxanthine increase, and we give artificial cell containing xanthine oxidase obtained from fermentation. And this is converted into uric acid that is excreted. The result reported in Lancet show that it's effective in decreasing hypoxanthine after 1 week. However, this is a very rare disease, so we're more interested in looking into a more common one, and the most common one is phenylketonuria. In this case, phenylalanine increased to toxic level. So we give artificial cell containing fermentation obtained phenylalanine ammonolyase. The phenylalanine is converted to cyanamic acid. This is converted by liver into hippuric acid and then excreted in the kidney. The result shows that normal animal has low phenylalanine level. PKU rat has very high level. Those on PKU diet has lower level. Those with oral artificial cell has level comparable to the normal rat. However, the enzyme is too costly. So as a result, we collaborate with IBEC company to produce recombinant enzyme is more practicable. And as a result of that, IBEC collaborate with a company in the US to prepare PEG cross-linked to the recombinant enzyme, and this has already been approved by FDA. Okay, let's move into the next topic, cancer therapy and immunotherapy. We can implant artificial cell into the body to work in next to biological normal body cell. For instance, we implant asparaginase artificial cell for lymphosarcoma, and the result shows that for those injected with control artificial cell with no enzyme, the number of animal having tumor continue to increase. In the case of those injected with artificial cell with enzyme, it only 50% of the animal show the tumor. These type of injection for tumor cancer does not require repeated long-term injection. So as a result, these are now being used in the form of PEG by another group for routine clinical use. We use artificial cell for the treatment of a skin cancer melanoma. This is based on nano-artificial cell containing nanobiotherapeutic. What we do is that we have hemoglobin and thyroxinase. We're crossing it together to form nanobiotherapeutic. We found that injecting this into melanoma mice normal can decrease the rate of growth but cannot suppress it. So we want to have something that can stay longer at the tumor injected site. So we put these into nano-artificial cell so it can sit at the site of action. This is just reported in press, should come out very shortly. We look at 2D culture, we look at 3D culture, we look at injection and the site and how reaction take place. "2D culture result, let's look at this very briefly. We look at proliferation, we look at entry into cell, showing that they enter into the tumor cell and resulting in a decrease in thyroxine level. We also find that they decrease attachment and they also decrease viability of tumor cell, decrease colonization of tumor cell, and decrease metastasis. In 3D culture, please refer to the article that just been published. It shows that thyroxine level is lower, cell count is lower, metastasis is lower, and cell cycle is also starting. We also study in very detail the different components of the artificial cell in affecting the growth of melanoma tumor in mice. You are invited to look at the article, but let's look at a key result. Results shown here. In a control group, giving anti-microcapsule or saline, the tumor continues to grow. But if you give these in nano-artificial cell, it suppresses the growth completely. And the animal body weight doesn't change and there's no side effect. Effect on the growth of high dose of melanoma tumor in mice. This is control. This is showing that with artificial cell, there's inhibition of tumor growth. And the body weight, the growth of animal is unchanged. We notice that occasionally there are some nodule in the nano-artificial cell. So the pathology look at this in more detail, and it shows that in the case of the control, the nodule are all neoplastic, 65% of tumor cell, whereas in the artificial cell one, the nodule, if present, are granuloma, foreign body type of reaction with no tumor cell. Furthermore, there are nanoparticles observed in the nano-artificial cell, none in the control group, and reactions are granuloma reaction. Cancer immunotherapy is a very popular way to also treat cancer. Like Lenker recently published article said that this has become an explosive area, but there are many problems that need to be solved, need for suitable delivery system. I just published an article reviewing the different configuration that you can easily use for cancer immunotherapy. Let's look at this. Let's look at artificial cell and drug delivery, example of some basic principles. There are different basic methods of preparing artificial cell. One of this is by immersion method to form artificial cell with the membrane. Another approach is nanobiotechnology based on the cross-linking of hemoglobin into polyhemoglobin. Another way is by conjugation, whereby hemoglobin is attached to polymer in the form of polyamine or dextran, or most importantly is polyethylene glycol, PEG. The original principle of preparing artificial cell has been extended by many groups. For instance, drug carrier first reported used polymer crossing protein using lipid membrane. Mueller first, and then Gregorius and Benham later used this method, modified it to use their lipid to form liposome. And importantly also surface modification with polylactic and also polyethylene glycol. Most recently, it was reported that PEG lipid nanoencapsulation of the mRNA for COVID-19 vaccine has been successfully used in the Pfizer vaccine. Biodegradable is another way to prepare these drug delivery systems. This was done back in 1976, but at that time this type of work wasn't that popular yet, and biodegradable at that time we used polylactic acid that is degraded into lactic acid in the body and become CO2 and water, so it's completely biodegradable. At that time I prepared nanoparticle or nanocapsule for enzyme or immunohormone vaccine and biology. This is an example using insulin. You can adjust the nanoparticle, capsule, or the microcapsule, microparticle to have different release rate from very slow to very quick or very intermediate. Vaccine, for instance, is one of the ways that was studied. And now you can use it using different polymer, PLA, PEG, PLA, PLGA, and others. You can make microcapsule, microparticle, nanoparticle, polymers, and different names. You can modify the surface to allow to target and other approaches. Another way in drug delivery was initially proposed is to put magnetic material inside artificial cell together with the biology. This way, it's now being used for drug delivery, it's being used in cancer therapy, it's used in laboratory analysis, it's used in bioreactors. Let's look at cancer therapy. One way is to make magnetic nanoparticle containing therapeutic agent or radioisotope and then put magnetic on the tumor site, and then they would attract the nanoparticle to the tumor site, resulting in localized therapy. And with all of these now, you can combine everything with different configuration of artificial cell, micron dimension, macro dimension for cell, nano dimension, or even nano biotechnological soluble complex of different forms. So as a result, the possible use of artificial cell for cancer therapy and cancer immunotherapy is unlimited and waiting to be thoroughly explored. Let's look at cell and stem cell therapy and regenerative medicine. Cell inside artificial cell is excluded from outside immunological cells or antibody and therefore can avoid rejection. Artificial cell containing cell now has four major groups of exploration: cells and tissue from the body, genetic engineering cells, microorganisms, stem cell. For instance, you can put hepatocyte liver cell inside artificial cell, and it's prevented from rejection. These are red hepatocyte in mice, quickly freeform rejected. If it's in artificial cell, it's not rejected. This is in artificial cell containing hepatocyte in gun-rate congenital hyperbilirubinemia. The level of bilirubin in the animal increased. When you inject the artificial cell, it slowly decreases back to normal. You can also put this using liver cell. The problem is that after injection, it only works for 1 year. After 1 year, the artificial cell is enclosed by fibrous tissue. How to solve the problem? Well, there are 4 ways. One way is to improve the method and also to improve the polymer material. For instance, the standard method results in sometimes imperfect membrane with exposure of the cell and weakening of the membrane also. We develop new method that can make a perfect membrane with no exposure or weakening of the membrane. Secondly, is to give it orally. We put artificial cell containing CH5 cell that when you give it by mouth can effectively convert hemoglobin, convert red urea into ammonia, which is then taken up by the same cell for protein synthesis and excreted in the feces, effectively lowering urea. And we also start using a more acceptable type of cell from yogurt. And as a result of that, it's now being extensively developed, especially by my PhD graduate, Prakash, into use for different areas extensively, and also by other centers. It works, but FDA does not allow for genetic engineering cell because they are concerned about its use. So, to avoid the use of this, I suggest to my group that we say look at something very safe. For instance, microorganism used in yogurt, lactobacillus. So we work on lactobacillus, show that you can do that. And because of this, it led to development of artificial cell containing related lactobacillus for use, for instance, in kidney failure patients. Kibo originally they want to collaborate with us, but we chose to do it with a Canadian group that did not come up with the funding to do the research. Another way is to remove cholesterol and other things by Package doing very well in this work. Another way is to look at regenerative medicine, because regenerative medicine only needs 4 months for liver regeneration. For instance, if you in animal study, if you resect the liver, removing 90% of the liver, if you then put artificial cell containing stem cell, the stem cell remove a hepatocyte growth factor that could go through the portal vein into the resected liver at high concentration. That will result in regeneration of the liver. For instance, this is one example of the result we obtained. In rats, we remove 90% of the liver, and the recovery of the rat is shown here. Rats with no liver removed recover no problem and live for the length of the study. Those with 90% liver removed, they die very quickly. Even the free stem cell does not work in this particular case because it doesn't stay in the peritoneal cavity to drain into the portal vein. 90% hepatomy with stem cell with artificial cell result in recovery of the animal. Now, artificial cell containing stem cell could be used, as we just discussed, to increase the recovery of the animal, and indeed the animal recover. At the same time, the liver also regenerate to the control-free and resect normal animal level, as shown here. The mechanism for this could be two. One is the implanted into peritoneal cavity, the hepatic growth factor is drained at high concentration into the portal vein, resulting in liver regeneration. Second mechanism is transdifferentiation into hepatocyte to carry out liver function. But this takes time, and sufficient transdifferentiation only happens in 2 weeks, so this cannot be the recovery of the 42 weeks. Most likely is for long-term possible use. So artificial cell containing stem cell could release hepatic growth hormone into portal vein, result in liver regeneration. However, injecting artificial cell into peritoneal cavity sometimes may cause peritonitis. So for practical reasons, I looked at the other possible thing of asking Dr. Liu to look into transplanting the artificial cell into spleen. This way, it's inside the spleen and function as a hybrid organ. And this way, the hepatic growth hormone is released into the hepatic portal vein and result in liver regeneration. And later, the stem cell transdifferentiate into liver cell and function. The spleen become a hybrid liver organ. Here shows that intraspleen implantation could result in the recovery of the animal and also liver regeneration to the normal animal. The fourth way to do this is to put cell in biodegradable form by experiment by Langer from MIT. We won't have time to go into detail. Now, let's look into biotechnology and nanobiotechnology. Recently, there's much interest in loading of red blood cells by reverse hemolysis, by putting in hypotonic solution, red blood cell membrane swell, release and content. At the same time, material from outside can go in. When they recover, the material is trapped inside. These are red blood cell membrane, therefore hybrid synthetic cell. Venter did some very exciting work whereby he used microinjection of synthetic genome into micron with the same content removed. The cell content is not there, just the membrane. And the result is replicating synthetic cell. This is very exciting. We have been able to convert waste like urea into essential amino acid using artificial cell with multi-enzyme cofactor recycling, using a rather complex system with recycling and conversion into L-leucine, L-valine, L-isoleucine. We can also put compartment inside artificial cell. You can see many compartments inside artificial cell to allow for intracellular compartment with multiple functions, stepwise function. In summary then, we can prepare artificial cell with different content. There's no limit. You can use many different types. And the artificial cell are made of artificial membrane or an artificial content, which could be biological or completely synthetic. And this has many possible applications, as we discussed. Giving you some examples, there are some agriculture industry, nanocomputer, nanorobotic, nanosensor that we don't have time to discuss, and these are also not my area. In my 1972 monograph on artificial cell, I emphasized that artificial cell is just an idea. Much physical configuration are possible. Indeed, in the last 10 years, there has been extensive increase in configuration as extension, improvement of the idea are being made. Within a few more years, it's expected that much more configuration would be possible as the area is extended and improved. I would like to acknowledge the support over the years by many granting agencies and many groups. I just need to acknowledge support of many years of research by different groups, and also the years of research staff that contribute to the present work. And they are the people that did all the work described here. Finally, may I invite you to visit the website. We don't have time to go into any detail. This is just a very brief summary. If you are interested, the detail in articles, videos, and books are available. If you have any question, I'll be happy to answer, and this is my email address.